So our next speaker is Dr. Rosalind Rabé, who leads the Greenhouse Entomology Research Program at Agriculture and Food Canada at Harrow Research Development Centre. Her group rigorously compares and contrasts the many new and old tools and agents used for crop protection, with an emphasis on the integration of new agents and technologies. Recently, the Greenhouse Entomology Program at Harrow has focused on better understanding of the impacts of artificial light on biological control agents and crop pests, as well as discovery, assessment, and potential commercialization of new biological control agents, including some native to North America. The research program ultimately aims to identify both the most effective and yet sustainable ways to manage diversity of pests present within Canada's greenhouse and vertically farmed crops. So, Rose, please take over.
Hi, Sheldon, you can hear me fine?
Yes.
Okay, I'm just going to switch over to the presentation view here.
Okay. There we go. Where we start. Thank you.
How's that?
Perfect.
All right.
Go ahead. Yes.
All right. So thank you, Sheldon. It's a real pleasure to be invited to speak today on the following all the research on the impacts of light on plant production. So obviously, fittingly, the next kind of piece of the puzzle is how are artificial lights going to affect the arthropods that are present in the growing environment. So this is research that we've been working on over the last few years. And of course, the premise for us to do this research all stems around the fact, the reality, that here in Canada, we have the reality of winter, which over the course of the Earth's yearly journey around the sun, we have a period of the year, which means that, the cold period of the year where we're further away from the sun overall, and we get overall less abundance of natural light that is delivered. And this is obviously contributing to a reduction in the potential for growing crops with natural lighting alone. So this is obviously a massive factor that influences crop production. And we also see that this is a major reason for the adoption of artificial lights in growing environments. And this on top of the increasing vertical farm acreage in the area in Canada in general. So these are the main drivers for today's use of artificial high pressure HPS lights, high pressure sodium lights, as well as LED lights in greenhouse and indoor crops. So we know that artificial lights can significantly improve crop production. Despite this, little research to date has been conducted to examine how new light environments actually impact greenhouse crop production as well. It influences both the good bugs and the bad. So we know that light impacts arthropods, insects and mites, such as mayflies. In southern Ontario, we have mayflies that are systematically drawn to the street lights every summer. And we also know that moths are attracted to flame. So clearly, these arthropods are responding to the light environment. And light is a very important factor for these organisms. But how important is it really to greenhouse pest management? So this is one of the big questions we've asked ourselves at the Harrow Research and Development Center over the years. And certainly we're only beginning to, I feel, understand really what are the interactions between the light environment and arthropods in these systems. So just to kind of put a little perspective on what we already know about the interaction between light environments and arthropods in the greenhouse. Well, actually, let's take a step back and look at arthropods in general. So here's the honeybee. This was the first subject of the first types of experiments that showed that insects actually do have some forms of color vision. And so this preliminary research done in 1914 was conducted by Nobel laureate Karl von Frisch. And he essentially did a series of experiments, classical conditioning experiments with honeybees. He took basically a little sugar, a dish of sugar water and placed it on a blue square in a grid, a kind of a checkerboard grid with other similarly shaped squares. But the rest of the squares were gray, various shades of gray in color. And what he ended up doing was through repeated exposures, the bee learned to actually recognize the blue color. And no matter where if one fish actually moved the blue square, the bee continued to follow and to note where the presence of the blue and the sugar water was present. Even when the dish was taken away, the bee went to the blue square. So this systematically showed us that bees were able to perceive light and color. He continued to do this type of experiment to show that bees were actually able to see white light, yellow light, blue light, and violet light. So this is a really important first step. And he also showed us for the first time that color perception of bees is somewhat like in humans, except that it's shifted away from the red. So whereas humans can see very well in the red range, we actually, bees are shifted towards the ultraviolet part of the spectrum. So bees actually cannot see, distinguish red from black, which is colorless to them. And it's also helpful to kind of compare the visual spectrums of the sensitivities of humans, plants, and insects on one particular graph. And this is just my very brief kind of rendition of my interpretation of the visual spectrums of each of these organisms, just so we have some kind of point of reference to compare these. Humans, for instance, if we started to look at the human visual spectrum, we know that humans can see light in ranging from violet to red, from 380 nanometers to 740 nanometers. However, they cannot see light in the UV range. So basically, there's a big barrier here to the perception of UV light for humans. In contrast, plants actually do also are highly sensitive to blue and red light. They actually, obviously these two LED light types are often used in growing environments, such as LED lights combining blue and red. And this is because of both chlorophylls A and B, and obviously carotenoids, and obviously I'm not the expert on this subject matter, but certainly all these photopigments absorb light within the red, violet, and then the blue areas respectively. In contrast, insects have a slightly divergent sensitivity to light. As we mentioned, most of the insects that we actually encounter in the greenhouse are bichromatic. So basically they are using two different photopigments receptors in the compound eye to actually absorb green and yellow light. Here we can see one peak on the green and yellow here, and then a second peak in the UV range. So obviously the perception of insects is quite different from that of humans and plants. So with this knowledge that we have on arthropods, we ask ourselves, so how does light quality, quantity, and daily duration affect the arthropods that we find in the greenhouse environment? More importantly, how does the light environment affect their populations and impact pest management? So many growers will comment, for instance, as some predators, Aureus insidiosus, for instance, are notoriously difficult to establish in the wintertime. And this obviously has important implications for crop protection as well. Ultimately, given the artificial lighting conditions we create to optimize food production, we also want to know which biocontrol agents should we be applying in various seasonal and lighting conditions, and how can we optimize these environments to improve their performance while minimizing minimizing the performance of pests. So one of the factors that is influenced by light in arthropods is diapause. So diapause basically consists of the response of an arthropod to a gradually decreasing amount of light per day. So as the photoperiod, and you can see on this graph here, so we are, for instance, in this season of fall, going from a photoperiod, we started like at 16 hours of light in the summer, and then are gradually going down as the season progresses into winter. And as we drop, the incidence of diapause is increasing for females of predators such as Aureus insidiosus. You could see that it goes up to 100%, close to a photoperiod of 10, 11, and 12 hours a day. So this is pretty dramatic. And as you can imagine, if this is a predator you're putting out on your crops, you would expect that there would be a decrease in its ability to perform as a predator. The side effects of diapods are that there's a reduction in rates of pest consumption, egg laying, and development. So obviously, if there is a pest that continues to thrive in the growing environment, is not necessarily sensitive to diapods, such as tropical species, this is a predator that's native to Canada, there would be a mismatch, and certainly pest management could falter in such a scenario. So in contrast, organisms such as Amblysea swirsky as a phytosayan mite comes from the Mediterranean basin, is not known to experience a cold, harsh winter such as Canada does, and doesn't diapause. So therefore, this kind of predator would kind of stick to a low level, to a non-diapause baseline foreseeably. However, our research is also showing that even non-diapausing organisms are sensitive to light and will be influenced by the light environment. So very interesting to study and compare these responses. So our research, as I mentioned, specifically sought to examine how a diversity of native and introduced arthropod biocontrol agents would fare when reared under simulated winter conditions relative to various artificial light and temperature environments, such as the ones that we would be creating in the growing conditions of the greenhouses or controlled environments. Our study subjects for these studies included the native predators Dicyphus hesperus, Aureus insidiosus, and as well as this, we also sought to study the aphelinid parasitoids of whiteflies and Carcia formosa and Eretmocerus aeromicus. And we also compared the response to light in multiple predatory phytosaids. So for each of the organisms, we use a specifically tailored experimental arena to contain and also to monitor the life history progress of each organism over time. And basically the trials that I'm presenting here were performed in controlled environments to really study the basic physiological response of these organisms to very artificial conditions that we create for these organisms. So these controlled environmental conditions that we compared included a simulated winter condition. So basically, this was our control, replicating a low temperature and a short day length condition. You can see, oops, sorry about that. Winter, so there was a colder temperature in this treatment as well as an HPS treatment, low light intensity, and a very short photo period of 8 hours a day. In contrast, we also had a simulated summer conditions, 24 degrees Celsius, higher light intensity of white light, as well as a long photo period of 16 hours. And we compared these to various LED treatments, single spectrum LED treatments, such as low intensity blue light. So all of these LED conditions were performed at 20 degrees Celsius. The exception was that there was a varying intensity of light delivered to the organisms, either 2.7 watts per meter square or 11 watts per meter square in the case of high intensity blue and red light. But each was delivered for 20 hours and no other light was exposed. So the insects were not, or arthropods were not exposed to any other light source at the time. And through these trials, we actually measured a number of environmental parameters, including developmental time, lifespan, and reproductive output. So amongst the first of our results from these controlled environmental trials, we first showed that the developmental time of generalist predator Dicyphus hesperus was virtually unaffected by any of the light treatments that we actually were testing. Actually, it was most affected, I'm sorry, I think I'm clicking the wrong button here. Most affected by the temperature. As you can see, developmental time was shortest under the summer conditions relative to any of the other conditions. This suggests that to us, in terms of developmental time, light is not as an important factor to this organism as is the temperature. However, when we look at the adults and factors such as longevity, this is a very different story. We see that the light environment actually considerably changes the response of organisms depending on the condition they are under. So with regards to the effects of light environment on this particular predator, Dicyphus, we observed that Dicyphus fared best under a high red light environment, high intensity red light, and it fared the worst under summer conditions. This is interesting as it suggests that the summer condition is actually shortening the lifespan of the predator. And it also very interesting to know that under HPS lights, female longevity was also boosted up. And this might be a response to the fact that HPS light has more full spectrum and is visible and could be triggering a better lifespan for this particular predator. We also notice, as with longevity, fecundity is also very much influenced by the light environment. We saw the best, the best predator fecundity under a low blue light environment, and the worst under summer conditions. So this means that over its whole lifespan, so since it's living a longer lifespan, it's laying more eggs over that period of time, and under a low blue environment, it is laying the most of all conditions. So when we look at these results, we certainly think that this could have an important impact on the persistence of a biocontrol agent on the crop over time. When we look at Aureus insidiosus and its response in fecundity and longevity to the supplemental lighting conditions or the artificial lighting conditions, we can see, again, a very big spectrum of response. If we look specifically at fecundity, we see that the predator performs best under a low blue light environment. It's actually, the females are actually laying on average 118 eggs over their course of their lifetime. And this is, represents nearly 3 times the amount that predators would actually experience under the summer conditions. This is a pretty big difference. And of course, there is a spectrum of other responses. So low blue lights certainly seem, and also low red light also seem to be supporting a good predator fecundity. Consistently red light and low red light, no matter what intensity of light, seem to be beneficial for this organism as well. In terms of the longevity as well, you can see, as we saw with Dicyphus hesperus, the predators fared best under the supplemental lighting conditions and the worst under the summer conditions. Moving on to the predatory mites, when we looked at Amblysia swarovski, Neocetus humerus, and Amblytromalis lammaticus for their response to these lighting environments, we can see again the response was quite variable. But again, if you, from a statistical perspective, the only difference we really noted was for the developmental time of Amblysia swarovski. And for this particular predator, surprisingly, because this is a Mediterranean species and should not diapause, its response in terms of developmental time, so basically it had the shortest developmental time under high red summer and HPS conditions. This was significantly better than what was observed under the other conditions. So certainly it seems that light, despite the fact that this is a non-diapausing species, can affect this organism in an important way as well. And it begs a bigger question. How is light affecting these species that supposedly should not be particularly sensitive to changes in light environments? Moving along to the aphelinid parasitoids of the whiteflies. Obviously, whiteflies are a dreaded pest in greenhouse crops, and Eretmoceras and Encarsia are often those that are applied to control these pests. When these predators were exposed to different lighting environments, it is surprising to note that the parasitism of whiteflies ranged considerably based on what species was observed. So we noticed right away that Eretmoceras in the blue showed a bigger variation in the response to light relative to Encarsia formosa. Now this is interesting as it never, this hasn't really been documented before. And you would expect that you'd have almost the same level of variation between the two species, but actually this is not quite the case. So perhaps there is something of biological significance that is influencing the response of these predators here to the conditions. Nonetheless, we showed that in the low intensity red LED light condition, we almost were able to achieve the same level of whitefly parasitism as was observed under the ideal summer conditions which achieved the highest level for this particular predator. In contrast, the other conditions seem to not really compensate for parasitism the way that that particular treatment did. So just to summarize some of the findings that we kind of come across for our biocontrol organisms in response to the light environments. So just some take-home points. What we found was, to reiterate what we've already stated, for hemipteran predators, it seems like summer conditions significantly reduce the developmental time, which is a positive thing if you're expecting a rapid population growth, but it also shortens the lifespan of the predator and lowers its fecundity, which is not necessarily a positive thing overall. We found in particular that a high intensity red light, LED light, improves male Dicephus longevity, and low intensity blue LED light actually improved the fecundity and longevity of both Aureus and Dicephus predators. For phytoseids, the general finding was that we found that only Amblyseius responded to the light environmental changes, and its development was fastest under HPS lights, next under summer, and then next finally under high intensity red light. This was significantly better than the other treatments compared. For whitefly parasitoid Moths eremicus, the summer condition also resulted in the greatest level of parasitism, but this was followed closely by the levels of parasitism achieved under low intensity red light conditions. This is all very well and good, but what does this all mean from a biocontrol perspective? So it is interesting to note that based on what we kind of suggested at the beginning of the talk, many insects do not have the photoreceptor or for red light. So basically, it is interesting to see that so many organisms, such as Dicephus males, which have a longer longevity under high intensity red light, and a higher lifetime fecundity for Aureus predators under both high and low intensity red lights. How are these organisms responding to the light if they cannot necessarily perceive it? Well, there's two possible ways that the light might be influencing these arthropods. The first is, of course, that as with new research is kind of showing slowly that it's not necessarily the perception of light that is important, but for this organism, the contrast of red objects or red light in contrast to other types of light or object colors is very important. This has been shown to be very important for Drosophila suzukii, for instance, which is specifically seeking out very ripe and red fruit, and that it could actually find and oviposit within it and feed upon it. So red can actually serve in this way as an attraction or a deterrent. Most likely what we are observing, however, is that arthropods on plants that are receiving red light are overall at an advantage. So healthy, growing, strong plants exposed to lots of red light, which is beneficial to the plants, is also beneficial to the organisms that are living upon them. And of course, all our trials were done with living plants. And so we can conclude that possibly what we're observing is this interaction between the positive influence on plants and a better nutrition for the insect that's living on it. What about blue light? Well, we certainly saw that high intensity blue light could have some negative impacts on the development of our predators, or shortening their lifespans, or having some negative effects on these organisms. But it seemed like at lower intensity lights, there was actually a beneficial effect of this wavelength on the organisms. So how do we explain this? From a physiological perspective, we know from research that high intensity blue light can actually be toxic to living things. If we compare a high blue light on the spectrum, the light spectrum, we see that it actually is right next to UV light. It's high energy, it has a short wavelength, and is therefore delivering more energy to its target than would other light spectrums, such as the red light, which is a lower energy type of light. So the study, a study in 2014 showed that on Drosophila melanogaster showed that blue light exposure in this particular species actually led to mortality of eggs, larvae, and pupae. And certainly as the intensity of the light is higher, this tends to be a more pronounced effect. So does this mean that too much light is a bad thing? Well, yeah, possibly. Just like UV light, it seems that blue light can be beneficial when it's applied at low intensities, but it's detrimental over a prolonged exposure. That said, a low intensity of blue light is probably going to be a beneficial thing, aspect to many arthropods. It may actually have an important physiological response in light of the incidence of diapause. For instance, lots of research is showing that exposure to blue light can disrupt diapause. This could be a way to actually modulate that physiological response in arthropods, and could be modulated to the benefit of pest management and crop protection. So just to summarize some of our findings, we can see from our research that the response of arthropods to light quality, to photoperiod, appear to be flexible in nature. It suggests that there is a lot of potential for modulating to create the right environment under artificial lights that will really optimize the organism that you're trying to target. However, our findings really clearly suggest that we're only beginning to address existing knowledge gaps in arthropod responses to light environments. We know that there are different and new types of lights that are surfacing every day. An example for that would be the new spectrum, Xuming's research on 24-hour lighting. We don't know how arthropods are going to respond to such an environment, and it would be very important to investigate that in the future. With all kinds of variables in the light environment, as the theory for how to apply the lights improves for crop production, we will be following in step, trying to figure out how these newly formulated crop environments are going to actually influence the arthropods. So lots of future research will be needed, and these will serve to really integrate these components to optimize crop protection in greenhouse and vertical farming environments. And obviously, it's very nice to study all of this in the confines of a controlled environment, but really the greenhouse is a wholly other ballpark, and lots more research will be needed to be conducted in that environment as well. So with that, I wanted to thank all the contributors to this research. I want to thank the Ontario Greenhouse Vegetable Growers who supported this research through the Agri Innovation Project 029, as well as Agriculture Canada and the individuals who have worked in our lab over the years, Dana Gagne, Dr. Yun Zhang, Jacob Basso, Erica Burton, Kay Puhaka, John Yang, and all the greenhouse crew at the services at Hera Research and Development Center. With that, I'd be happy to take any questions if I had any time to do so.
Thank you, Rose. Very nice discussion. Sort of helps with everything that's been going on with the other speakers that went on earlier, but shows us lots more work to be done because you've got some interesting stuff coming through. There's one question that I have for you here. It's from Mike. And any sense that fecundity would increase in biological control as generations become conditioned to the greenhouse conditions over time, i.e. the lab-raised bios could be conditioned to lab conditions and perhaps perform more negatively in the greenhouse environment conditions. Comment on that?
Okay, so just so I understand correctly, so it is the fecundity of predators may adapt to the lighting conditions of the greenhouse better than they do to the conditions of the cabinets, right? Is that?
Yeah, or the other way to look at it is you're raising these in a controlled environment to cabinet and under the lighting, artificial lighting conditions. When you put them in the greenhouse, would there be a change? Would there be
yes,
the original light spectrum, and they won't be able to perform the greenhouse? So that's a
yeah, that's an excellent question. So because obviously the controlled environments are really pushing the limits, creating very artificial environments to study the physiological response of these organisms. But in reality, in the growing environment, we don't have just blue LED lighting. We also have natural lighting. And of course, that's another aspect of the research that we've done in the past. And if I had more time, I would also discuss that as well. And of course, there's an important interplay between the two lights. So absolutely, there could be this aspect where the supplemental light is extending the photoperiod and creating an overall a much better condition for oviposition that we could be observing for the predators. Definitely, future research is needed in that to confirm.
Thank you. I don't have any other questions for you, Rose, at the moment, but there's another one. Do the different light spectrums affect the effectiveness of the beneficial insects?
Yeah, so obviously it does. The light spectrum, obviously, and I'm not sure if this is referring to light spectra outside of the ones that we've considered, but certainly while we've already shown that you know, a pure white light environment is beneficial in the sense that it's very much visible by arthropods, and it's going to trigger an important biological response. It is certainly going to, white light contains blue light and red and everything else, so will trigger that those responses in those organisms. There will certainly be shifts away. If we're talking about red light, we may not see the benefit readily for arthropods. That being said, we don't know exactly if there are other ways that the organisms are responding to. So lots of future research to investigate this.
All right. Similarly, David asked, that was a question from Vincent, a question from David. Other than diapause, could predatory longevity be dependent on food quality as a result of plant response to light?
Yes, and this is where I need to pull in the physiologist here, because obviously I've spoken to Shuming several times, because I thought it would be so neat to actually look at the pollen production on crops. And I think, Shallon, we've had this discussion a million times. Yes. How does pollen production, which is an important source of food for predators, how does it change in the crop over time? And is it going to be a significant factor in the establishment of predators? And of course, yes, the pests may also be influenced by the light environment. And so there's this interplay between the food that you get from the pest and the food you get from the plant.
Thank you. A question from Henry. Would a local species of Orius do better during the summertime?
Oh, that is a good question. I mean, certainly it's a fight or flight strategy. It's with the local species, you can't help but to think that they have one shot at reproduction, and they are just going to give it all that they have to be successful as a species. So it is quite possible, and I would actually love to compare Orius strains from tropical climes to those of the indigenous Canadian species to see just how different they are in their reproductive strategies. That's a great question.
Good, excellent. So that's, you got a lot of work coming up.
Yes, we need help.